I begin this video by telling you, you are infected. Yes, you really are infected. Ah! I will explain what I meant by that at the end of this video. It's a way to keep you watching. And you get bonus points if you can ID what movie that image comes from. Now, we're going to talk about the structure of living things. And we're going to start at the very beginning, because in my mind, there's no reason to hide anything. This is simple. The Legos that build everything in the universe and every living thing are three basic particles: protons, which are positively charged subatomic particles; neutrons, which are neutral subatomic particles; and electrons, which are negatively charged subatomic particles. Protons and neutrons come together in the middle of atoms to form a nucleus, and electrons are found orbiting them, but they don't fly in circular orbits, they fly in three-dimensional orbitals. So what's an orbital? An orbital is a probability map of where the electron spends its time. So if we look at this 1s orbital here, this is a cloud, and the electron zooms around in that cloud and can be found anywhere in that cloud. It's a density map of where it can be. There are different shapes and sizes of these orbitals, and they're dictated by quantum mechanics. We are only going to talk about s orbitals and p orbitals, because most of the elements, the atoms that make up life, will have their electrons in these two types of orbitals. S orbitals are spherical, and p orbitals have this dumbbell kind of shape, and there's three of them that are perpendicular to one another. You can see here's a p1 orbital, a p2 orbital, and a p3 orbital. You will also have different energy levels, and we're just going to call them 1, 2, and 3. It's more complex in quantum mechanics, and if you really want to go explore that, knock yourself out, but that's beyond what we're going to talk about in this class. Level 1 can only have an s orbital, level 2 can have an s or p orbitals, and level 3 can have an s, a p, or a d orbital, but we're really not going to talk about d orbitals because they get even more complex and difficult to visualize. So just think about these orbitals, and these are the places where electrons can hang out. So those are the subatomic particles. These are put together to form atoms, and how you put them together dictates what atom is formed. If you take one proton and you put in one electron, that electron will go in the lowest energy orbital, which is the 1s orbital, and it will zoom around that proton. That is a hydrogen atom. If you take two protons and you add two neutrons and two electrons, the electrons will again go to the lowest energy orbital, that is the 1s orbital, and they'll both be in there and it will be full. When an orbital is full, it will be very non-reactive. So helium is actually one of these unique gases that is non-reactive. To look at some more examples, if you have 6 protons and 6 neutrons and 6 electrons, that makes a carbon atom, and it will fill the 1s shell here in the center with 2 electrons, the 2s shell with another 2 electrons, and then it will put 1 electron each in the p orbitals. So it has 2 p orbitals. It will fill each one of them individually before it puts another electron in. So those are the rules. So you have 1 electron in each of these orbitals. In the case of carbon, you have 2 p orbitals with 1 electron each. These orbitals are half full, and therefore carbon wants to fill these orbitals by finding 2 more electrons. In the case of oxygen, you have 8 protons and 8 neutrons and 8 electrons. So again, you'll fill the 1s orbital, you'll fill the 2s orbital, so that's 4 electrons, then you'll put 1 electron each in the 2 p orbitals, and then finally you'll put a second electron into one of the p orbitals. So you'll have 2 orbitals again, that are empty, and oxygen will also want 2 more electrons to fill those orbitals. These molecules, like hydrogen and carbon and oxygen, will always be searching to see if they can find electrons to fill those orbitals by pairing with other molecules. Before I leave orbitals behind, I need to mention an amazing little trick that carbon atoms pull off. They can rearrange their orbitals, and instead of making 3 p orbitals, they make 4 sp orbitals, as shown here. You can see the little sp orbitals. They look different, and there's 4 of them arranged in a pyramid, and each will contain its own electron. This gives carbon the capability of forming 4 bonds instead of 2. Because of this versatility, carbon serves as the backbone of organic chemicals, those that are used in living systems. There are over 118 known elements. How many are used in living organisms? Surprisingly, only about 10 are used at concentrations greater than 1%. If I take your body, remove all the water, and look at what atoms are left, I will find that carbon, nitrogen, oxygen, phosphorus, hydrogen, and sulfur account for 97% of your mass. If I then include iron, magnesium, calcium, and potassium, the number rises to 99%. Think about that. You are composed of only 10 different atoms. What about that last 1%? This contains various unusual elements that are used in very specific purposes in your body. The atoms that compose living things do not have full orbitals, and all of them want to share electrons with other atoms. For example, hydrogen gas has 1 electron in its s orbital. It wants to share 1 electron with somebody else. An easy thing to do is to find another hydrogen atom and make a hybrid orbital where there's 2 electrons in it, and that makes both of the hydrogen atoms happy. And in fact, this is how hydrogen exists in nature. This electron sharing is called a covalent bond, which I'll often just refer to as a bond. For other elements, carbon forms 4 bonds, oxygen forms 2 bonds, and nitrogen forms 3 bonds. Phosphorus and sulfur are unique cases that can bond in a number of different ways. As another example, oxygen has 2 half-full p orbitals and is searching for 2 more electrons. It can pair with 2 hydrogens, and these hybrid orbitals now share the electron between them. The chemical formula is H2O, and yep, we are talking about water. There's one more important property that I want to introduce now since I'm talking about water. Atoms want their electrons to be near their nucleus. In the case of water, this means that the oxygen, because it has more protons, will pull the electrons away from the hydrogen. This leaves the poor hydrogen starving for electrons, and they will be attracted to other molecules nearby, especially if those molecules have a high electron density. This attraction is called hydrogen bonding. It's very important in biology, and we will come back to it in other lectures. And a molecule that shares electrons unequally is a polar molecule. Many molecules do not share well and are polar. The property of polarity is also important in many functions in biology. Going forward, it's going to become very tiresome to continually draw all these orbitals or space-filling molecules, as shown here for the water molecule. So instead, we're going to use a little bit of a shorthand. And that's how a number of these other structures are drawn. Each element is drawn within a letter code that corresponds to its code in the periodic table. Then you will draw little lines in between the bond to other elements. So in this case, we have methane here, we have a carbon, and we've then drawn a bond to the hydrogen here. It would be tiresome to continually draw all these orbitals, so scientists have come up with a shorthand for drawing molecular structures. Each element is represented by a 1 or 2 letter code, and these codes are found in the periodic table. And then the bonds between them are drawn as lines. For example, hydrogen gas, as shown here, is drawn as 2 H's with a line in between them to represent the bond. You can also draw semi-3D representations, as shown here for methane gas. We have a carbon and then bonds drawn to each of them, and a bond's thicker coming towards you and kind of thinner going away from you. And methane is actually a carbon and 4 hydrogens. Methane gas is the major component in natural gas that you burn in your home. It is more common to draw things flat. The last example shows one type of fat molecule found in butter is what's called a triglyceride, and triglycerides are what actually make up fat. The 3 carbons form glycerol, and attached to it are 3 fatty acids. So you have the carbon backbone right here, and then you have 3 fatty acids. Fatty acids contain a long chain of carbons and a carbon and 2 oxygens at the end, right here. This is called a carboxylic acid. These long chains of carbon share electrons equally, and they behave in the exact opposite way of water. So the electrons are all equally shared. In fact, water does not like to associate with these non-polar molecules. And you can see this in your everyday life if you mix oil, a non-polar fatty substance similar to butter, and water. The mixture will separate into 2 layers. So, now you know way more about the atoms of life than you ever thought you would. You're welcome! Let's move on to monomers. The atoms of life can get together in all sorts of ways, just like you can build almost anything with Legos. However, living things, for all their diversity in shape and sizes, only use a few assemblies of atoms, called molecules, to make everything. These are carbohydrates, nucleic acids, amino acids, and lipids. They are the monomers that make up the polymers of biology. Carbohydrates are molecules that have carbon, hydrogen, oxygen in a ratio of 1 to 2 to 1. If you arrange that, that becomes CH2O, and you can now see how carbohydrates get their name. They're a carbon, carbo, and a hydrate, water. As an example of a carbohydrate, I show the structure of fructose, a sugar that you'll find in lots of different soft drinks and other foods. Oh, and by the way, scientists are, again, lazy. They get tired of drawing all those carbons. So in many types of drawings that you see, and by the way, scientists are lazy. They get tired of drawing all those carbons in organic molecules. So they don't. If you see a bond, meaning a change in direction in a structural drawing, as shown here, and there is no letter there, it is assumed to be a carbon. Nucleic acids are the monomers that make up the polymers DNA and RNA. And they are involved in storing and expressing our heredity. They have 3 parts: a phosphate, a sugar called ribose, and a base. And in this case, I'm using the example of adenine. Lipids have a similar structure to the triglycerides that we just talked about. They contain a glycerol backbone of 3 carbons with an oxygen off of each end. 2 of these glycerol carbons are attached to fatty acids and are nonpolar. The 3rd position on the glycerol background will have a polar group on it. This difference in polarity is essential, as you will see in a minute, to the function that lipids have. Finally, we have amino acids, which you may have read a little bit about if you are interested in fitness, especially weightlifting. Amino acids have a carbon, called the alpha carbon, that is attached to the nitrogen, the amino group, and a carboxylic acid, COOH. Thus the name amino acid. They also have a variable group designated R. This variable group dictates the unique properties of the amino acid. These 4 monomers that we just talked about will link up to form 3 common polymers: polysaccharides, proteins, and nucleic acids. Polymers can be very simple. As an example, starch contains long strings of just 1 monomer, glucose, hooked together. Polymers can also be very complex. Proteins have unique sequences of amino acids, and this specific sequence dictates their function. This is where the diversity of life is expressed. As a specific sequence of monomers put into polymers changes, it changes the property of the polymer, both in how it functions and how it looks. Let's take a minute to look at just a few more details about biological polymers. Proteins are made of amino acids. There are 20 different amino acids that are commonly found in proteins, and their chemical structures are shown here on the right. The side chains, the R groups, for each amino acid dictate their properties. Some are charged, acidic and basic amino acids, and like water, they are hydrophilic, while others have no charge and lots of carbon and hydrogen, for example, the aromatic and the aliphatic amino acids. These structures share electrons well, are nonpolar, and don't like water. They are similar to the oily compounds that we talked about before with lipids, and this dislike of water is called hydrophobic. Amino acids are put into long sequences, polymers, to make proteins. The sequence of amino acids in a protein dictates its function. One example here is I'm showing you a space-filling model of the enzyme nitrogenase. Nitrogenase is involved in converting nitrogen gas into ammonia and is a very important biological process. Proteins can be enzymes, such as nitrogenase, that speed up chemical reactions in cells, and they can also serve a structural role. The two other polymers found in every cell are DNA and RNA. Each is made up of 4 nucleotides. For DNA, it's adenine, A, thymine, T, cytosine, C, and guanine, G. RNA is similar, but uracil, U, substitutes for thymine. Because of the properties of the bases, A can form 2 hydrogen bonds with T, or U, and G can form 3 hydrogen bonds with C. This is called a base pair, and the size of chromosomes is described in a number of base pairs. A does not form hydrogen bonds with C, and G does not form hydrogen bonds with T. DNA and RNA will polymerize in long strands where the ribose of one nucleic acid will bond to the phosphate of the next nucleic acid. And you can see this over on the side here, and you have these long strings, and this shows a string of DNA going along and being linked up into a long polymer. DNA forms the hereditary material and has a second strand as a backup. This second strand is the exact complement of the first strand, having a complementary base that will hydrogen bond with the other strand. In this way, complementary strands can recognize each other and zip together to form polymers millions of base pairs long. DNA that is millions of base pairs long makes for a long, skinny molecule. In the case of the bacterium Escherichia coli, its chromosome is about 4.6 million base pairs. The length of this molecule is 1.4 millimeters, yet E. coli has to pack that into a cell that is only a few micrometers, that's 1/1,000,000th of a meter, long. Obviously, DNA has to be highly organized. There are all sorts of proteins in cells that help package and organize the DNA. And in bacteria, this is kept in an area called a nucleoid. And you can see this here on the right is a electron micrograph of the E. coli bacterium, and the clear areas is where the nucleoid is. Unlike in eukaryotes, the nucleoid is not separated from the rest of the cell by a membrane. The next molecule that I want to talk about that's really important is ribosomes. Ribosomes are assembly of mostly RNA and some protein. The picture at the right is a space-filling model of what a ribosome looks like. You can see all the RNA, cyan and off-white in this picture, and the proteins that help carry out its function, red and purple. You have 2 subunits to the ribosome. There's the 30S subunit, or small subunit, and there is the 50S, or large subunit. Ribosomes are the protein factories of the cell. They translate the instructions copied from the DNA into RNA and take that and translate that RNA into protein. We'll have much more to say about this process in another video.
Lipids have two sides to them: a water-loving head, called hydrophilic, and water-hating tail, and this is called hydrophobic. As I explained earlier in the video, this is because of their polarity. One part of the glycerol has a polar or charged group on it, and the other part of the glycerol has two fatty acids on them that don't like water and are hydrophobic. If you put a large number of lipids into a glass of water, the water will repel the hydrophobic portions, and these will associate to get away, just like oil does. This self-assembly ends up forming a lipid bilayer that in living cells is referred to as a membrane. Membranes in cells are made of lipids, but also contain proteins, some of which may contain polysaccharides. The lipids on each surface of the membrane can flow around one another, so membranes tend to be fluid structures when functioning properly. And because of the proteins decorate in, it's called a fluid mosaic model.
Membranes are a permeability barrier, but this is not absolute. Gases, such as carbon dioxide, oxygen, and nitrogen gas, can pass through pretty easily and pretty quickly. Small uncharged molecules, such as water, also pass through pretty easily. However, sugars such as glucose and sucrose, or charged elements such as chlorine, potassium, and sodium, rarely cross a membrane. If a cell wants to move these in, it has to have proteins that recognize them and move them through. Obviously, things larger than glucose and sucrose, such as proteins and other polymers, are unable on their own to diffuse across the membrane, and the cell has to have very special functions to actually get these across the membrane. The lipid composition of a membrane affects its permeability and stability. First, let me explain the difference between a saturated and an unsaturated fatty acid. A fatty acid with a long chain of carbons with no double bonds in it is saturated. It contains as many hydrogens as it possibly can. Our example here is palmitic acid. The carbon chain on palmitic acid will be straight and can associate very tightly with its neighbors. If a fatty acid has double bonds in it, where two neighboring carbons share two sets of electrons, that's called a double bond, it is called unsaturated. In this case, the fatty acid will have a kink in it, and this type of lipid with unsaturated fatty acids cannot pack as tightly. The amount of unsaturated fatty acids in the membrane has two consequences. It makes the membrane more permeable, and it makes the membrane more fluid. Membrane fluidity is critical to the function of the membrane. As the temperature of the environment drops, membranes become less fluid, and at enough temperatures, they will freeze, halting their function and ceasing cell growth. This is one of the reasons refrigerators preserve foods. In summary, membranes perform three functions. They have a selective permeability that keeps the cell components in and the environment out, and they serve as an anchor for proteins that carry out various functions for the cell, such as transporting molecules in and out, moving the cell around, and generating energy. Before we leave the cell cytoplasm, I want to stress one more thing. Bacterial cells are not big bags of fluid with DNA, ribosomes, and proteins floating around in them. They are tightly packed with very little free space. It is so tightly packed that most of the water inside the cell is bound up to the various macromolecules of the cell. The image shown here is my cheesy little mock-up of how you should think about the cell. It's just really packed in. You can see here proteins and ribosomes and the membrane and DNA all packed together. Now let's take a step back for a second. Most bacteria are very small, and you may wonder why. Being small has important advantages. When you are small, you have a higher surface to volume ratio, meaning you have more surface to the amount of volume that you have. For example, a 1 micron cell with a 1 micron radius has a surface to volume ratio of 3. This means you have more surface interacting with the environment and thus find it easier to bind to and absorb nutrients. If you make just a tiny change, you increase the radius by 1 micron, the surface to volume ratio halves. Having more volume also makes it harder to get macromolecules from one area to the cell to another, which will slow down growth and likely put you at a disadvantage versus other bacteria that are smaller. Now we move on to the outside of the cell to talk about the cell wall. Most bacteria live in environments that are far more dilute than their cytoplasm. Nature abhors this type of disequilibrium because there's much more solutes on the inside of the cell than the outside of the cell. However, diffusion of the solutes inside of the cell is not possible since the membrane prevents it. Instead, water moves into the cell to try and balance things out, and this creates pressure, which is called osmotic pressure, as more molecules crowd into the cell and the cell swells and will eventually burst. To prevent the cell from bursting, most bacteria have a cell wall, pictured here as this brown little circle, and the cell wall holds that membrane together and prevents lysis. Most bacterial cells can be divided into two groups, gram-positive or gram-negative. This grouping is based upon their staining properties. The structure of these two cell wall types is quite different, but they do share a unique polymer that is found only in bacteria, peptidoglycan. Peptidoglycan is formed of two sugars, N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM. These two polymers polymerize together end to end to form long chains, and each chain is then cross-linked to other chains by peptide interbridges made of amino acids. So you can see where the name peptidoglycan comes from, because it contains peptides and it contains these sugar glycan molecules. If you look at this, you'll see here's this long series of polysaccharide, and then it's cross-linked with these NAM, in between these NAM molecules, with a peptide interbridge. If you look at this in 3 dimensions, it's shown over here on the right, and you'll see it's this mesh structure, and that mesh surrounds the cell and holds the membrane together. The cell wall, or more accurately, its synthesis, is also critically important to treating disease. Humans don't have a cell wall, nor any of the enzymes that synthesize it. Thus, any drug that inhibits peptidoglycan synthesis is likely going to be of medical importance. About 50% of the antibiotics prescribed in the US are actually cell wall inhibitors, and this graphic shows you a number of different drugs that are used. Cycloserine is a molecule that inhibits the formation of this D-alanine dimer. Right? Alanine is amino acid, and part of the synthesis of peptidoglycan is making this D-alanine dimer, and if you can't make that, you can't make the cell wall. Bacitracin, shown here, blocks the flipping of this carrier molecule called bactoprenol from the outside of the membrane to the inside. Bactoprenol has a NAM-NAG monomer added onto it, and then it flips back to the outside, and this NAM-NAG monomer with the peptide interbridge is what actually gets built into the growing chain of peptidoglycan. If you can't flip it back, that inhibits the cell and prevents peptidoglycan synthesis. And finally, probably the most commonly prescribed cell wall inhibitors are penicillin and ampicillin and other what are called beta-lactam antibiotics. These inhibit the enzymes that link the NAM-NAG polysaccharide chains together. They inhibit the enzymes that synthesize or link those peptide interbridges, and these are called transpeptidases. Vancomycin also blocks the linking of these peptide interbridges, but in a different way. It forms a cap on top of the peptide interbridge so that the enzyme can't get to it and link them together. We now move on to quickly describe the two cell wall types. Gram-positive cells simply have a thick layer of peptidoglycan outside their cellular membrane. And that's it. It's just this thick layer of peptidoglycan, and you can see that here. Gram-negative cells have a more complex cell wall composed of 2 layers. A thin peptidoglycan layer, and then outside of this, an outer membrane. So gram-negative cells have 2 membranes, the cytoplasmic membrane and the outer membrane, which is part of its cell wall. The outer membrane has some unique properties. First, it is not a permeability barrier. Scattered throughout the outer membrane are protein tubes called porins. And these porins allow small molecules to pass through to the cytoplasmic membrane. Secondly, the surface of the outer membrane has a unique molecule, lipopolysaccharide, or LPS. LPS is composed of a lipid part, lipid A, and this is attached to a long polysaccharide that projects into the environment. LPS forms a barrier for the cell that slows down the diffusion of hydrophobic molecules toward it. The presence of LPS in the outer membrane makes gram-negative bacteria more resistant to some antibiotics and many disinfectants in comparison to gram-positive cells. This is, of course, species dependent. Some species will have more resistance. Some species that are gram-negative will have less of resistance. Outside the cell wall, there can be other layers. One of these can be a capsule. Capsules are polysaccharides or proteins that envelop the cell in a protective blanket. You can see 2 examples here, both showing bacteria surrounded by their outer capsule. The image on the right shows Streptococcus pyogenes binding to a tonsil cell. S. pyogenes is the cause of strep throat. Capsules serve the cell by helping it to retain water. Capsules slow evaporation of water away from the cell, as protection either from predators or from destruction by a host's immune system, and for attaching to surfaces. Capsules are sticky and can glue cells to surfaces. And this is shown again on the picture on the right, where you see the capsule helping the bacterium stick to a tonsil cell. Bacterial cells can also have rod-like projection structures outside the cell called flagella, pili, and fimbriae. Flagella are organs of motility and help to move cells through liquids. Pili can have several functions. They can help to exchange DNA between cells, or they can serve as organs of motility. And finally, pili and fimbriae can also serve to attach cells to surfaces and to protect against predation. You can imagine with all these fimbriae sticking out, it would make it more difficult for a predator to consume a bacterium. Now to explain that statement at the beginning of this video. You may be wondering how you can be healthy and still have every one of your cells infected. To understand this, we need to spend a little time on eukaryotic cell structure. This is your cell structure. These are how your cells are built. First of all, eukaryotes are more complex. Eukaryotic cells are about 10 times bigger than bacteria. They also organize the cytoplasm into identifiable units, such as a nucleus, an endoplasmic reticulum, what a cool sounding word, a lysosome, a Golgi complex, and mitochondria. You probably learned all about this in your biology class. I want to focus here on the mitochondria. The mitochondria are the powerhouse of the cell. There are numerous and can account for 20% of the cell volume. They take all the food that you eat and convert it into energy and reducing power that powers your body. If you look at the structure of mitochondria, you start to notice some odd things. They have 2 membranes, with the main energy generating reactions occurring in the inner membrane. Does that remind you of anything that we just discussed? That's right, they resemble gram-negative bacteria. Is it possible that mitochondria have an ancestor that was a bacterium? It turns out it's true. As further proof, mitochondria have their own piece of circular DNA. They also contain ribosomes that resemble bacterial ribosomes. if you determine the sequence of the RNAs that make up the mitochondrial ribosome and you compare those to all living things, you find the mitochondria are closely related to bacteria. That's right, every one of your cells has mitochondria in it. And those mitochondria are ancestors of bacteria. In the far distant past, the ancestor of the mitochondria was engulfed by another unicellular cell. But instead of being eaten, it formed a partnership with its host cell. It converted ingested nutrients into energy, and in return, the host cell gave it protection. What I am talking about is the birth of eukaryotic cells billions of years ago. We now reach the end of the lecture on bacterial structure. The take-home messages I want you to get out of this are the following. Atoms are made of the three particles. Atoms are made of just three particles: protons, neutrons, and electrons. Atoms form molecules, and the cell is made up of just a few types of monomers: sugars, nucleic acids, lipids, and amino acids. These monomers form the polymers, polysaccharides, DNA, RNA, and proteins. These polymers come together to form all the structures of the cell. Things like membranes, chromosomes, ribosomes, and cell walls. All right, that's it. Have a great day.